And we're very, very pleased to have this exciting talk tonight on how and why biological shapes change during evolution. A history of ideas from D'Arcy Thompson to developmental genetics. What a wonderful title, what a wonderful spread in history and, of course, in biology. And it is to be given by Dr. Arkhat Abzhanov, who is a reader in evolution and developmental genetics at Imperial College London and researcher at the Natural History Museum, also in London. And it's good, really good to see that these two organizations, one a research establishment with a vast collection and to a public appeal, and the other an academic institution with good, fine research. And these two things link together to show the unity of such organizations. And it must be great for our speaker tonight to be able to work in both of those organizations. Arkhat is interested in a variety of topics related to the vertebrate craniofacial or head development, head genetic conditions in humans, and cranial evolution. The animals he studies, and this is really rather a especially interesting aspect, range from the laboratory model species, as you might call it, such as chicken embryos and mouse mutants, to the non-model species from the wild, such as Darwin's finches, as we'll see, I'm sure, some pictures of later on, crocodilians, and bats. Please, can you give a very warm welcome to our speaker tonight?
Thank you. Thank you, Paul, for the introduction. Thank you all for coming. It's a pleasure and honor to be here today. My lecture will be focusing on ideas which were inspired by great thinkers such as Darwin and D'Arcy Thompson a long time ago. And hopefully I will show you how their ideas are still very relevant and how they continue to be a source of inspiration for our current research. I'm fascinated with biological shapes. So you look at any living object and you can just marvel how complex and beautiful they are. And I'm trying to understand how you build such shapes and how they come into being, how they evolve. So these are drawings from Ernst Haeckel's 19th century paintings, which show some of this diversity. You see they come in soft forms, they can be hard bodied. There are these beautiful patterns of diversity, beautiful shapes. These are faces of New World bats. We actually have a project on these. Again, wonderful diversity. So the two questions which I'd like to address today. The first one is, what's the origin of any biological shape, such as this head of the bird with a beak? As you know, that it's very rare that shapes in biological shapes come in isolation. They usually come in these patterns. Closely related species have similar yet unique shapes. So what's the origin of such diversity? How is this diversity generated? It turns out there are two answers which are highly related. One answer, of course, is evolution. If you think about what happened during the evolution of what we call a modern bird, this quail on the right, it evolved from a very simple single-cell animal, and this evolution started about 500 million years ago. During the Cambrian one, it evolved through a series of successive steps. It first evolved the internal skeleton, it evolved two pairs of limbs, it evolved a head with jaws, it evolved eyes, it evolved a tail, it evolved feathers, and a beak, and then turned into what we call a modern bird. That's one answer. There's another answer. The same bird is a product of another process, what we call individual development. It starts with a very simple structure, a single egg, a fertilized egg, which then, through a series of steps, develops into what we call a modern bird. That is, it develops an internal skeleton, it develops two pairs of limbs, it develops a head with jaws, it develops eyes, it develops feathers, it develops a beak, and it hatches into a bird. So these two processes, evolution and development, so the question we would like to this connection between the two, between the survival of the fittest, the evolutionary process, and the survival of the fittest, is what we're trying to understand, because we feel that the breakthroughs in both evolutionary biology and developmental genetics can be mutually very beneficial and can help us understand both. It will help us to interpret the data from mouse, for example, mutants, and chicken embryos towards biomedical purposes, but also better understanding of developmental genetics, how the genes build tissues and how tissues build organs, can help us understand evolution as well. In fact, here's a quote from Darwin himself where he said that in the eyes of most naturalists, the structure of an embryo is even more important for classification than that of an adult. For the embryo is the animal in its less modified state, and it's so far it reveals the structure of its progenitor. In fact, for Darwin, for his evolutionary theory, it was one of the foundations, one of the pillars of his theory, in addition to the biogeography, in addition to the fossil record. Embryology, developmental biology of the time, was very important to him to understand evolution. In my own research, I focus on what we call craniofacial development, that is the origin of the entire head and face region in the vertebrate animal, where it turns, so this early embryo, the faces are very similar in early stages, and they turn this recognizable heads of the birds. This is a chicken embryo, or the mammals. This is a mouse embryo. It's a very, very complicated process with many different cells and many different tissues, such as the brain, the skull bones, and the cartilages, and the muscles have to interact to produce this very complicated structure. So when the mistakes do happen, it can lead then to craniofacial abnormalities. And in fact, over a third of genetic conditions in humans have craniofacial abnormalities. So it's a very large biomedical issue, understanding how development of the head normally works and what happens when something goes wrong, because there's so many genes involved and so many cells. But we also understand that it's the changes, it's the tweaking of the same program that builds the head and the face that must be behind this diversity. And diversity is stunning, where you can recognize a particular species just by looking at its head and face, a picture. And you can also tell what this particular animal is doing for a living ecologically, because a lot of these features that you see in the face are very adaptive as well. So we have a range of projects, as mentioned, which include projects on birds. All the projects today I'll be talking about will be case studies on birds, but we have similar studies on other types of organisms as well, such as crocodilians, other reptiles, bats, and other mammals as well. So there are three types of approaches which we take, which are colored here in different, indicated here with different colors. The blue ones, these are methods which are aimed to understand what we call morphology, the biological shape itself. And these methods can range from simple methods, such as counting segments in the millipede, to much more complicated three-dimensional morphological analysis using computers, using so-called 3D morphometry, when we look at the entire skull and its complexity in 3 dimensions in the computer to understand exactly what happened during evolution. The other set of methods here shown in orange are the methods of comparative embryology, when you look at key species to understand what actually is different between them, how you make the face differently, what are the interesting genetic changes which correlate with these changes in the biological shapes. And finally, these methods here in green, they're functional methods where we take these genes, when we modify them, we change them in order to understand how these genetic changes, whether they are causative of these changes in biological shapes. So we link the two causatively. And this continues on and on. It's kind of the gathering of knowledge. So much of the work that I'll be describing today is inspired by this book. Last year, in 2017, we celebrated exactly 100 years since publication of this book called On Growth and Form, by famous British mathematician and biologist D'Arcy Thompson. Here you can see that Nature, when the book came out, Nature magazine wrote in this book that this book, On Growth and Form, at once substantial and stately, is to the credit of the British science an achievement for its distinguished author to be proud of. It is like one of Darwin's books, well considered, patiently wrought out, learned, and cautious, a disclosure of the scientific spirit. This is the first edition here, published by Cambridge University, and this is one of the more recent editions. This book continues to inspire us biologists 100 years on, and I'll tell you why, but this book is actually very important for our understanding of how biological shapes, again, can be measured and described, but also how they can change. D'Arcy Thompson both looked at evolutionary side of things. So he famously put the skulls in the grid, and he compared biological shapes, such as skulls, for example, of this primitive horse that turns into a modern horse. And he looked at this, how it's warping, how the shapes are warping from the basal, from the kind of the ancestral to the modern state, inventing single-handedly what we call morphospace, the space within which biological shapes can be understood. So morphological evolutionary transformations, which he applied, these geometric transformations, he applied the knowledge of geometry and mathematics to understand biological shapes. Recently, I gave a talk at the St. Andrews University, where D'Arcy Thompson worked for very many years, and this is the book which I was shown in the library. That's the book which was given to D'Arcy Thompson. It says here, to D'Arcy Thompson from his friend Henry Osborne, who was a very famous mammologist who studied mammals, January 1911. And you can see here on the pages of this book, hopefully you can make out this faint pencil drawings. These are grids, which you can see that this is a book from the personal library of D'Arcy Thompson that he's applying, he's beginning to apply his mathematics to understanding biological shapes. Another important breakthrough that he made, he was able to compare biological shapes during development. So these are, for example, embryonic, these are fetal skull shapes for the chimpanzee and the human, and he looks at how these biological shapes change geometrically during development to understand what kind of changes are taking place, and he's able to compare them directly. So geometric transformations of biological shapes, he writes in his book, constitute a proof that variation has proceeded on definite and orderly lines, that a comprehensive law of growth has pervaded the whole structure of integrity. So the law of growth is something that became very important to him. What we now know about this process is that the genes are important, information in the genome, and so is the environment. And it's this machinery, all these developmental genes, which interpret both of these sources of information, and then they work and they work out all the codes, and they build the cells, they build the tissues and organs, and build us from scratch that actually that make all the diversity. And he believed that it is these laws of growth which were describing these developmental processes, which were not just important. To D'Arcy Thompson, they were key. They were front and center in terms of explaining, in his mind, how diversity came about. He gave a lecture in Edinburgh in 1894, which he called Some Difficulties in Darwinism for the British Association meeting. And this lecture was never published as an article or a book. And the Nature magazine correspondent writes here about his speech, that D'Arcy Thompson doubts the efficacy of the struggle for existence in the case of hummingbirds, etc. And in these cases, he regards the profusion of forms, colors, and other modifications as due merely to laws of growth, and thinks that growth may be more exuberant in the absence of struggle and hardship. He basically thought that all that natural selection, all the screens, may not be enough to explain how diversity comes about in the first place. And he believed that it's the changes in this underlying the laws of growth, the developmental principles, that must be the generative force that makes all the diversity. That put him at odds with mainstream Darwinism at the time. But the question still stands, and you'll see that the relative significance of extrinsic, that is ecological forces, and intrinsic ones, that is genetic, we're still trying to figure out the right answer. So here I'm going to introduce a concept called adaptive radiation. So adaptive radiation is the rapid evolution of morphological and ecologically diverse species from a single ancestor, when a single ancestor very quickly, burst-like almost, gives rise to many very distinct progeny. It usually implies two coincidental processes. One is multiplication of species number, which is referred to as species richness, how many species a lineage produces, but also increased phenotypic disparity, which we refer to as morphological diversification, how different these descendant species are from each other and from the ancestor. Such evolutionary success is often attributed to a key innovation, a novel breakthrough trait, that is some kind of novelty. It could be morphological trait, for example, a beak or wings, or it could be even behavioral trait that promotes this kind of radiation and diversification of the whole group of species. And classical examples of adaptive radiations are Darwin's finches on the Galapagos Islands, the Hawaiian honeycreepers in Hawaii, the cichlid fishes in the Great Lakes of Africa, anomalous lizards in the Caribbean, marsupials in Australia, and the list goes on, because this process is extremely important in producing variation in the living world. So one example that I'll be focusing on of adaptive radiation is Darwin's finches. So here's a diversity shown here. There are 18 or so species, each of which is unique in terms of its beak shape and its diet. So they all evolved from a single ancestor which landed in Galapagos some time ago. And now the beaks are different, and again, they use them for very different diets. So here's a large ground finch. It's using its very deep and very broad bill to crack very hard seeds. Here's a cactus finch. It is using its pointed bill to get into the cactus flowers and fruit to get the pulp and the nectar. Here's the vegetarian finch, as the name suggests. It has this very, very narrow, very deep, but very narrow bill, which is using sort of like garden shears to cut young leaves and eat them. A very unique diet and adaptation on the islands. Here is the woodpecker finch, which has the very straight, very strong bill, and it woodpecks around for insects, and occasionally, famously, when it finds the larva deep inside the hole, it will make it a really little tool from a twig, take it out from the hole and eat it. So again, each of these species is different, and the story of Darwin's finches, the reason why they bear Darwin's name, begins with a trip that Darwin took as a young man. He was in his early 20s on board HMS Beagle. And he writes here, this is in the Voyage of the Beagle, this is some 20 years before the origin of species. Here he describes these birds, the Darwin's finches, that the most curious fact is the perfect gradation in the size of the beaks in the different species of Geospiza. This is one of the major groups with the Darwin's finches. From one as large as that of a hawfinch to that of a chaffinch and even to that of a warbler. So that he's referring to the English species here describing Darwin's finches. Seeing this gradation and diversity of structure in one small, intimately related group of birds, one might really fancy that from an original paucity of birds in this archipelago, one species had been taken and modified for different ends. You can see here that, again, 20 years before the origin, the big book, he's already thinking about Darwin's finches in the right terms as the ants arrived there and through adaptive processes gave rise to this entire group. So here is what we call a phylogeny, genealogy of all the species from a single ancestor which came from most likely from South America or the Caribbean and gave rise to all this beautiful diversity of tree finches which feed on insects and beetles and warbler finches which feed on flying insects and ground finches which feed on seeds, and here is the famous vampire finch. Great diversity. We think of the bills as tools. This is a drawing by Peter and Rosemary Grant, where he, again, they directly compare different beak shapes to different tools, which they use masterfully to get on very different food types. They also reported, the Grants, Peter and Rosemary, this is a very long-standing collaboration, Peter and Rosemary, they're two professors, a couple, which work at Princeton University, and they've been studying Darwin's finches and their ecology and their evolution for over 40 years now, and Galapagos going back there every year. They reported that the Darwin's finches, when they hatch out from an egg, they already have the right beaks on them, suggesting that everything that produces the beak is genetic and it's developmental. It's happening inside the egg. In fact, if you look inside the egg and the embryo, this is still several days before hatching. You can see that they're already developing the right beaks, the right beak shapes. Here is the cactus finch. It's developing very long pointed bill. And this is the ground finches. They're already developing very deep and very broad bills. Again, it's the process, as you can see, it's independent of the usage. It's fully genetic and it's developmental. And that's exactly where we come in because we'd like to combine these development genetic approaches to evolutionary biology to understand the kind of more mechanistic, to get more mechanistic answers. But as D'Arcy Thompson famously said, if you do not understand how the organism grows, that can be, or the organ grows, you do not understand the organism. So the bill, or the beak, the bird beak, the way how it grows is it begins from this potato-shaped structure. It's very bulbous, and it has this very large group of cells inside, which are proliferating, which are dividing all the time, so-called growth zone. And as the beak grows, this growth zone begins to decay, and it decays at a constant rate until it decays into nothing. And that's actually what generates the beak shape, because it generates the tip. And the beak shape, mathematically then, is something what mathematicians would refer to as the envelope. That is all the shapes, the summary of all the shapes of the growth zones which existed during development of this structure. And we worked to mathematically model this. We looked at not only the mechanisms, exactly how the cells divide and how they produce the bill, but also we tried to capture the shape of the bill and mathematically. And we showed that D'Arcy Thompson, for example, who believed that something called affine transformations, scaling and shear, that's a particular type of mathematical transformation which he used to compare all these biological shapes in his book. We applied them and they worked beautifully on these Darwin's finch bills, suggesting that the mathematical tools that he was using were absolutely correct. And in fact, the beak shapes are not changing at random here. The beak shapes here are related to each other mathematically. This does mean that in order to understand the mechanisms, genetic mechanisms for these biological shapes, we have to look at the embryos. And this means that we have to, for this project, to go to Galapagos. We have a particular strategy in place because it's known that the songbirds, once they come into the nest, for example, a particular species lays 6 eggs. If you let them lay 6 eggs and you take the last one, the female then lays a replacement egg because she knows how many eggs she wants to lay. And then she begins to sit in the clutch and incubate the eggs. So if you take that one last egg, so then you can have this particular embryo to look at its look at the genes and the cells inside to understand how it grows. So this is the pictures from the field. Some of the nests are easy to find. Others are a little bit more challenging, and they like to build them on top of this very large cactuses, as you can see here. Then the embryo is taken to the incubator. This can be at the station if we're on one of the main islands, or we camp out and we have an equipment tent. We have a generator and we have an incubator and the coolers and the freezers in the field. So the way it works, again, we collect an egg, we incubate the embryos for precise periods of time because we know exactly how the embryo should look like at different stages, and that allows us also to compare different stages and corresponding embryonic stages from different species to each other. So you can see here, these are the embryos from the large ground, medium ground, large tree, and vegetarian finches. So we can compare the big shapes. We can collect this tissue and look at the genes inside, look at the activity, look when they're active and what they're doing inside. For example, this is a summary. We looked at many genes. I'll show you the story of 2 such genes. So here is an ancestor. We believe that all the Darwin's finches evolved from a bird similar to modern-day grass squid, which still inhabits South America, Central America, and the Caribbean. These are the 3 species representing some of the diversity. The sharp-billed finch, the large ground finch, and the cactus finch. And one gene which we found is called BMP4, which stands for bone morphogenic protein, which means this particular gene is known to actually control bone development. In this particular gene, we found that it's activated in very early stages, much earlier and much stronger than other species, specifically in the embryos of the ground finches, the finches which have this bullfinch-like bills. Another gene, called calmodeling, or CALM for short, was expressed or active in a new domain, kind of in a new fashion, very strong new domain in the cactus finch, in species which have these elongated bills. And the question you may ask, are these molecular changes which we detect, these correlations, important? So here's a slide which advertises our model system. One of our model systems is the chicken embryo. Why? Because we can easily obtain them, we can incubate them, we can make this window on the developing chicken egg, and we can experiment with this embryo directly. We can move the tissues around, we can do different surgeries, we can activate or downregulate particular genes. That's exactly what we're trying to do here when we target the phase and we upregulate or downregulate the genes which are normally active in the developing phase. So here, for example, we'd like to understand whether BMP4 gene, which is activated early and at very strong levels in the ground finches, in these bullfinches, whether that change is enough to produce a meaningful biological change in the embryo itself. So here's a laid embryo of a normal chicken, which has a rather pointed bill because it's designed to peck on small seeds on the ground. So in this embryo, this is experimental embryo, we upregulated, we activated BMP4 to very closely mimic the changes which we observe in embryos of ground finches. That is, we upregulated this gene at earlier stages and in much stronger levels. And if you do that, these bills will transform into much deeper and much broader bills, which are very reminiscent of the bullfinch bills in the Darwin's finches. In fact, if you carefully titer, we used specially designed virus to deliver this molecule, this gene, activated inside the developing beaks. And if we carefully titer this, if you control the levels of BMP4, then it goes from a normal what we call wild type chicken bill shape to something that looks like a small ground finch or the medium ground finch, or even the large ground finch, which have this very deep bill which grows from the forehead and very, very broad bill that is, again, similar to what you see in the ground finches in the wild. But that leaves us with an interesting dilemma. That is, all the species use the same what we call structural genes. The genes which encode for muscle and bone and cartilage are the same. All the birds have the same genes. In fact, we all, as vertebrates, we have the same genes that control formation of the bone and cartilage. all of them are using the same regulatory genes. BMP4 is a regulatory gene, but BMP4, all the species have BMP4. We have BMP4. So how do you use the same structural genes and the same regulatory genes, which are already in the genome, and produce this diversity? So the best analogy I could find to explain what we're observing is architecture. When we use the same building materials, we use cement, we use bricks, we use wood, and we use the same tools, and yet we build very different looking buildings. So it's really the blueprint, how, when, and where we use the tools differently that makes all the difference. So we're looking for, essentially, in our quest, we're looking for this blueprint that, that allows these animals to use their mutations, natural killer mutations, to use these regulatory genes differently in different species, and that's actually what makes a huge difference in the result. But here's an interesting additional question, which is, why then some groups of animals are much more diverse than others? Again, if we use the same architectural analogy, then you see that the scapes here, the cityscapes, are, for some groups, are very diverse, and for others, they're much more boring. So in other ways, so why Darwin's finches are so diverse? So Charles Darwin, when he, again, The Voyage of the Beagle, when he described the species, he wrote that the remaining land birds, now he's referring to Darwin's finches, form a more singular group of finches related to each other in the structure of the beaks, short tails, and form of body and plumage. There are 13 species which Mr. Gould has divided into 4 subgroups. All the species are peculiar to this archipelago. So he already recognized that they're quite peculiar, they're quite different from all the other birds. And he was absolutely correct. Now, so here you're looking at the diversity of Darwin's finches. I included some of their Caribbean relatives here to show that their beak shapes are extremely diverse, and they can feed on insects and fruit and seeds and even leaves here. The only other group of birds which diversified on Galapagos are called Galapagos Mockingbirds. So they evolved from a single ancestor, and they evolved as 4 species which are subtly different from each other. They're very similar to each other, but they're different a little bit in plumage. You can recognize them. And here is a related species from the Caribbean, an endemic from the Caribbean. As you can see, that the diversity here is far greater for Darwin's finches than for any other bird, including the mockingbirds. So the mainstream explanation for Darwin's finches diversity is adaptationist. So the canonical adaptationist explanation for radiations such as Darwin's finches, which was developed by an evolution biologist, George Simpson, in the 1950s, involved simultaneous divergence of multiple lineages as a consequence of entering new, what we call adaptive zones, new islands, or finding new niches of foods, these ecological opportunities. So the reasoning goes that the ancestor arrived here. These are all new islands, completely open, new habitats, new food types. Then it jumps to another island, evolves a new species, and through island jumping and perhaps moving around, the entire complement of species would evolve with time. The problem with this reasoning is that if you look at other species living on the same Galapagos Islands, it doesn't really explain what's happening there. That is, so these are 2 papers. The timeline here runs from the modern to about 3.5 million years ago here. Here you can see Darwin's finches arriving about 2.5 million years ago. They're not the first colonists here. The mockingbirds arrived there about 1 million years earlier, and the doves arrived early as well. Here's an even more informative slide from another paper, which shows that this is modern times. This is about 4 million years ago. The Darwin's finches here, they arrive, again, also about this Darwin's finches here, about between 2 and 3 million years ago. They're not the first colonists. The mockingbirds arrived much earlier. For most species, and the Galapagos are populated by many other land birds. For many species, they didn't even evolve endemics. For example, the vermilion flycatchers or the cuckoo birds look identical. In fact, it's the same species as in the mainland. The mockingbirds, as I mentioned, evolved 4 species, and the Darwin's finches uniquely evolved multiple species, all different looking from each other. In fact, if you look at the summary of the colonization of the Galapagos Islands, you'll find that there are 28 endemic species and 6 endemic subspecies on the Galapagos archipelago, which evolved from 18 different ancestral bird species, which arrived from different directions to colonize the islands over the past 45 million years. Only one of these ancestors, only one of these colonies diversified and gave rise to radiation. and that's the Darwin's finches. Why? To address this question, we're using this novel computer-based methods. This one is called BAM. It stands for Bayesian Analysis of Microevolutionary Mixtures. It's really a computer analysis of different traits and different evolutionary trajectories across these different birds to understand the rate shifts in speciation and diversification, basically how many species these different lineages produce and how different they become. We can test the relative significance of intrinsic, that is morphogenetic, and extrinsic, that is environmental factors. We can also identify the triggers and modifiers of such evolutionary radiations. We also take advantage of a comprehensive avian database, which was just recently assembled by our colleagues, that includes 9 morphological traits. That is everything that ornithologists care to measure in birds. That is different beak parameters, wing, leg, body weight, etc., and 6 environmental factors, their diets, which habitats they inhabit, territoriality, whether they live on islands or not. And this is what we're trying to use to understand across the birds what are the relative significance of this environmental versus genetic factors. So the first step in our analysis, which we just finished recently, is to understand this one family called Thropidae, or tanagers. It's a very large, it's the largest songbird family, very diverse, second largest avian family, with 400 species of tanagers. And once we analyze this, the idea would be to use the same approach and then to understand all the songbirds, all the what's called passeriformes, all the songbirds, passerines, with about 6,000 species, and then move onward to all the birds, which number about 10,000 species in total. So if you look at just this one family of tanagers, again, 400 species, a very diverse family. If you look at this particular phylogeny here, shows you the speciation rate, how many species they were able to produce, and you can see there was a lot of speciation. The colors here reflect kind of the warm colors, the red and orange, reflect high levels of speciation, and the blue ones reflect very low steady state levels of speciation. You can see there's a lot of speciation early on in the history of this family, and then it cooled off. So there was very little speciation going on, except there were 2 interesting clades, 2 interesting groups, which completely changed that trend. One of them is the Darwin's finches, and some of their immediate relatives in the Caribbean. So that you can see here, this red spot indicates a upshift in speciation rates. Another one is called seed eaters, or Sporophila, and I'll come back to this later. It's a very interesting group where a lot of species were produced, but they all look rather alike, a very different radiation. Now, so I don't expect you to see the details here, but I'll just give you the main messages. What we're trying to do here, remember I told you that we're looking at the changes in things like different traits, such as tails and the wings and the beaks. And we found that for all the traits, here's the tarsus or the leg length, here's looking at the wing length, here's looking at the tail. What you can see within this family that there are massive changes happening early in evolution, and then this entire part of the family then inherits the change that early ancestors produced a long time ago, and that's pretty much it. So you can see that the changes later in evolution of this family are very minor, and they don't lead to this massive changes. Kind of the different color shows that there was a kind of massive change in the tail length, for example, here. So this is also true for the different parameters of the beak. Here is the bill depth, here is the bill width. So you can see this early changes in evolution, and then very few changes in everybody else, except there's only one exception here, and this is the Darwin's finches. So if you zoom into Darwin's finches, what you find, curiously, is that the changes in the bill shapes, such as changes in the bill length or the bill depth, happen very rapidly. They happen in very closely related species and happen very frequently. So this is something that really makes them different from, in all the parameters, only beak shape is doing that. So that's the only parameter that we looked at that is changing in ways that correlates with this speciation uptick in Darwin's finches. So there's a recent paper by our colleagues which looked at the bill shape across all the birds. This is the phylogeny or the genealogy of all the birds. And they found that for all these bird lineages, they found that there was an early uptick and change in the bill shape, and then followed by this cooling off and steady state change over time. So there's a lot of experimentation early on, and then it cools off. And that's exactly what we see for everybody within the tanagers, except, again, Darwin's finches. So this is something we call the morphospace. In the blue is the beak shapes. This is a geometric space inhabited by different beak shapes The blue are all the tanagers, the entire family. The pink here are the Darwin's finches, and the blue here are that other radiation, the Sprophila, the seed eaters. And you can see that a very large chunk of this morphous space is occupied by the Darwin's finches. And another important thing is that they're centrally located. So this is where morphology begins. They can change along different dimensions. They can increase their length, they can increase their depth, they can shorten the beak, they can elongate the beak, and so forth. Why is that important? Well, because if you look at the diet morphous space, the diet diversity, again, the diets which are consumed from insects to worms to flowers to seeds and leaves by all the family, you can see the diversity of the diets for the Darwin's finches is enormous. Essentially, it matches almost the entire diversity of the diets for the entire family, whereas the Sprophila, even though they are quite even more numerous in terms of species number, they are only feeding on seeds, and their diet diversity is very, very low. So Darwin's finches, because they were able to change in different dimensions, in different directions, they're much more successful in consuming very different diets. Now, here's another morphous space which compares directly how the Darwin's finches here, the morphous space, changed. So these are the Darwin's finches here. The green one is actually, they're very diverse relatives in the Caribbean. And you can see they're quite diverse. Whereas the Galapagos Mockingbirds, they originated from a diverse mockingbird family on mainland. But since they arrived to Galapagos, they did not change very much. They did change clearly to adapt to the new conditions. The beak shapes are distinct from the rest of the mockingbirds, but they didn't experiment at all with their beak shapes, and they basically just, they just enjoyed that particular new beak shape, but did not experiment much. This is very different from Darwin's finches, and of course, Darwin's finches not only they changed their beak morphology, they changed the species number, whereas the Galapagos mockingbirds, since they arrived, if you look at the Galapagos mockingbirds within the Galapagos family, the mockingbird family, you'll see that they did not change their speciation rates at all. They just arrived there, they changed somewhat, and that was it. And that's basically how all the birds behave on Galapagos. The only group that is different is the Darwin's finches. So the environmental explanation then, the adaptation explanation, does not really explain why Darwin's finches are so diverse. So the alternative explanation, a more structuralist interpretation, which was proposed by Kevin Burns in his 2014 paper, in evolution says that the ancestor of all these birds perhaps possessed a developmental genetic architecture passed on to its descendants that included a greater variety of regulatory genes controlling nasocranial development. That is, perhaps what the real innovation they have is a change in the developmental program that allows them to build, because probably you have more genes or something, that allows them to build more diverse beak shapes. Now we're beginning to check this hypothesis. For example, this is the phylogeny which indicates that Darwin's finches, shown here, are part of a larger clade, part of a larger group called Caribbeanae. These are the relatives which mostly live in the Caribbean. And if you look at different beak parameters, for example, how the build length changes and the build depth, what you'll notice is that within this clade, back in the Caribbean, this entire story of build diversity begins. So it's not really Darwin's finches, but also their close relatives in the Caribbean, this particular group here, the closest relatives in the Caribbean, that are beginning to change very rapidly. And this is where the story begins, back in the Caribbean. So what we did, we looked at the genes, and we found that this genetic story which controls, the genetic program that controls beak shapes in Darwin's finches, which involves the genes, multiple genes controlling the cartilage shape and the bone shape, and this is a very complicated modular system in place, is also found in the close relatives in the Caribbean, whereas the more basal species, that is species which are not changing very much, not very diverse, they have a much simpler development program in place. This is the same genetic program that we see, for example, in chicken embryos. And it's a program that doesn't allow really for much diversity because there are very few genes involved, so all these dimensions not necessarily built by the same genes. Here in Darwin's finches, different dimensions, for example, the length is regulated by one gene, the depth is regulated by another gene, so it's a much more flexible program. This leads to a very exciting concept called evolvability. So evolvability is the degree to which a biological system can evolve into a diversity of adaptive solutions to future environments. Two organisms with the same phenotype, the same shape, and fitness in the current environment may differ in their evolvability owing to differences in the cryptic evolutionary potential of their genomes. Basically what it's saying is that these colonies, which arrived to Galapagos, were not equally evolvable. Some of them had something in their genomes, something in their genetic programming that allowed them to produce much more variation much more quickly, and therefore adapt to the new resources and new conditions much more rapidly. So what we're doing right now is we're looking at Darwin's finches. We're looking at all the genes which produce different beak shapes in hopes of understanding what we call the network of genes which controls, it's a very complicated regulatory network which controls different parameters of the beak shapes in Darwin's finches. Then we would like to go back to the Caribbean and understand exactly how this network, this is what we believe is the true innovation here, this new development network which they invented back in the Caribbean, and the ancestor of Darwin's finches already came fully endowed with this capacity to produce all the variation. We'd like to understand how it happened back in the Caribbean, how this program actually evolved. Another textbook story of adaptive variation, which I mentioned early on, is called the Hawaiian Honeycreepers. This is an older clade. The ancestor of Hawaiian honeycreepers came to Hawaii about 10 to 15 million years ago, so they evolved even greater variation. They evolved to become, they're famous for this very long sickle-shaped bills, which they use to suck on the nectar inside the flowers. And you can see that the diversity is stunning. The recent analysis showed that they evolved from rose finches, from the Asian rose finches. And in fact, the basal-most species of the honeycreepers are still finches, such as the Nihoa finch, and they feed on seeds. Then many of them became creepers. They became insect feeders, and independently, some of these creepers evolved to become honeycreepers, and again, they evolved these beautiful long bills with particular shaped skulls that allow them to feed on the nectar from these flowers. Just like the Darwin's finches, there is something very special about the group of birds on Hawaii. So the Hawaii were colonized over a longer period of time from Asia. There were 25 or more bird colonization events on Hawaii over about 10 million years. Out of 71 endemic bird species which are known to have existed on Hawaii, 42 species, well more than half of all the endemic species which ever evolved on Hawaii of the birds were Hawaiian honeycreepers. So you can see here diversity of honeycreepers. This is the only other radiation that the birds showed on Hawaii. These are called Hawaiian thrushes. They evolved 5 species. One of them, unfortunately, is now extinct. As you can see, they evolved 5 species on main islands, but they're very similar to each other, and they're very similar to their ancestor, whereas the honeycreepers are very different from each other, and they're very different from their ancestor. So really, what we wanted to do is to compare what is so special about these two groups, what makes this diversity in Hawaiian honeycreepers, and are there the same mechanisms which make Darwin's finches very diverse? To understand this question, we wanted to understand what happened with their skulls, not just the beaks, but the entire skull, because the entire skull is important for proper biomechanics of the beak. So we put these landmarks on, these are computer tomography scans, high quality, high definition computer tomography scans, which allow you a very accurate 3D representation of the entire skull. So we put these landmarks, which computer then recognizes when computer program compares the shapes in 3D. And we also recognize that the skull will be very modular. That is, there will be functionally different parts of the skull. For example, the beak, the orbit, the part of the skull which protects the brain, and so forth. And we wanted to see how these modules behave during evolution as well. So what we've done, we scanned skulls from all the Darwin's finches and the relatives, all the honeycreepers and the relatives, but also what we call outgroups. These are more ancestral shapes. All of them are, as you can see, all of them are seed eaters. So these are all small finches, which have very similar beaks. And that's the result. This is the morphospace. Morphospace inhabited by different skull shapes. This is the ancestral morphospace for the small finches. As you can see, they're very similar to each other. These are the Darwin's finches, which are much more diverse in all directions. But it's the honeycreepers which show that not only they match the diversity of the Darwin's finches, but because they evolved this long sickle-shaped bills and novel skull shapes, they evolved this novel, they occupy this new parts of the morphospace, and they kind of broke through even further. So one thing which emerged from this, I remember I showed you this slide, which shows different modules. We wanted to see how these different modules, the beak and the orbits and the skull vault, how they change relative to each other. So here's a table. The closer you're to 1 for example, here's the beak relative to the rest of the skull. If you're at 1, that means your beak changes the exact same way as the rest of the skull. So it means that it's highly integrated with the rest of the skull. The lower the number, that means the more flexible that structure, for example, the beak is from the rest of the skull, so it becomes more what we call modular. And as you expect, Darwin's finch bills are more modular than many of the other structures, but it's the Hawaiian honeycreepers which showed incredibly low numbers of integration for all the other parts of the skull, suggesting that it's really all these different parts that we care to compare. They somehow turned their skull into essentially a Lego set, when they can change all the different parts independently with great flexibility in different directions very rapidly. And this is what we believe explains much of their diversity. So that's something that they basically took another step. We believe it's still developmental. We still believe that it's the developmental program that controls integration of the different skull parts during development that is being affected here. And they somehow, we still don't understand the mechanisms. We'd like to know how this normally occurs in normal birds, like, for example, chicken embryos, and how they manage to change that so that they significantly lower the integration of the skull parts, increase modularity, and allow and generate all the beautiful diversity. So moving forward, what we're doing right now, so beginning to look at the rest of the songbirds. So we used the tanager family as kind of a testing ground. We'd like to look at all the songbirds. So this is looking at all the songbird species, 6,000 species in all, just looking at speciation rates. Again, just looking at how many species are produced. As you can see here, there was an early experimentation with speciation, then it cooled off for most of the lineages. There are interesting exceptions here shown in red. These are recent radiation events. Darwin's finches are here. This is the Sporophylla, another interesting group within the tanagers. You can see the corvids here, and here's the white eyes, the zosterops. These are the famous white eyes which kind of colonized the Indian Ocean. One interesting observation is that it is believed, again, if you open the biology textbook, that living on islands, speciation islands, somehow special, and then because it somehow aids speciation. Most of the stories which we detect using our methodology in songbirds, they're shown here in green, are mainland speciation events. These are the things like tyrant birds or the oven birds or the spine tails. Some of them are island speciation stories, such as the honeycreepers and the Hawaiian honeycreepers and Darwin's finches, but most of them, curiously enough, tend to be mainland stories. And we would like now to understand what is common about all these radiation stories, what is driving them, with the hypothesis, again, that is its internal mechanisms, developmental programs here, which have been altered, and they increase the availability of some of these groups, which allow them to back the trend of exploring early on and cooling off speciation and diversification over time. So there's something very special about these groups. There are about 7 or 8 groups which recently managed to somehow break through these constraints and allow them to produce a lot of variation very rapidly. So to conclude, there are several clades of passerines which showed recent rapid increase in speciation rates. A minority of these are island taxa, which by itself is quite curious. Darwin's finches plus closely related, these are the relatives from the Caribbean, show exceptional levels of both morphological, cranial diversity, and speciation rates. And these phenomena are not easily explained by the purely adaptationist explanations and require a more structuralist approach, or at least a combination of the two. So we're exploring now the relative roles of both extrinsic, that is ecological, and intrinsic, that is morphogenetic factors using a combination of phylogenetic, morphometric, and developmental biology methods. And our analysis now so far show that an intrinsic innovation at the level of craniofacial developmental program is what's likely increased evolvability of Darwin's finches and fueled their morphological diversification. And this is what, in addition to this, all ecological opportunities, which we believe must be an explanation for their great diversity. As a postscriptum, Darwin himself, if you read his works, especially the late editions of The Origin of Species and his letters to his colleagues, he comes back to this idea of laws of growth, because the developmental biology, embryology continued to thrive, and he continued to pay very close attention to it, and he begins to recognize, later in his life, the significance of this new field. He writes here that we may easily err in attributing importance to characters, and in believing that they have been developed through natural selection. We must by no means overlook the effects of the complex laws of growth. We thus see that many morphological changes may be attributed to the laws of growth and the interaction of parts independently of natural selection. And it has been an error on my part and a misfortune to me that I did not largely discuss what I mean by laws of growth at an early period in some of my books. He's here referring to the early editions of The Origin of Species. And finally, endless other changes of structure in successive species may, I believe, be accounted for by various complex laws of growth. So that actually helps us to reconcile the two great thinkers, Charles Darwin and D'Arcy Thompson, in ways that their approaches are actually complementary. The idea that the adaptationist environmental explanation, the environment changes drive evolution is very important, but so is the genetic underpinning, and the ability to produce variation, the ability to produce novel forms, novel biological shapes appears to be very critical as well. So here I'd like to stop, thank my members of my group, in particular, my PhD student Ashley Rini, whose work I showed quite heavily today, and my postdoc Maria Dobreva. I'd like to thank all of other members of my group, all of our field assistants, many of our collaborators, including Peter and Rosemary Grant, and many other important collaborators, all of our funding sources, and of course, you for your attention. Thank you.